It's a pleasure to be here, to see many old friends. And I would like to thank the organizer, of course, for kindly inviting me. Everything I'm going to talk is based on the Gaussian distribution. We're going to have a nice discussion. So this is, I think, one of the most classical papers I like from Tom Mistelli, showing how the genome is organized. So we have lots of talks on the helix, nucleosome formation, and then chromatin loops, we had some. We're going to focus in my talk on the chromosomal territories, okay? The largest scale over there. And is this one? No. Okay. So we're going to talk on, the question we ask is, can we relate genomic structure and function, the structure to function? And to do that, more importantly, you know, all the literature up until recent years has been focusing on this four-letter alphabet to interpret the genomic behavior in general. And we would like to see whether we can have a physical basis based on interloci gene topology to be able to interpret differential expression of genes in different types of cells. So that is and we're going to do this using the Gaussian network model. And the Gaussian network model has been actually introduced more than 2-3 decades ago for proteins or biomolecules. We applied to the ribosome, we applied to entire viruses. Now the question is, can we adapt, can we use this type of network model to examine chromatin dynamics? So we're going to use high-C maps, and we're going to transform them into what is called Kirchhoff matrix, connectivity matrix. And I'm going to talk about that, some eigenvalue decomposition to extract the dominant features. And then we would like to look at the square fluctuations of loci. What's the loci? This is about, let's say, each locus is about 50 kilobase long. So we are talking about a totally different scale, not molecular at all, which requires, you know, a few parameter readjustment, new assumptions, but that's what we've done. So Gaussian network model, I'm not sure how familiar you are. Some of you are, I'm sure, but maybe not everybody. So, and since there was lots of discussions on that, let me give you a few, just quick background. Now, we started again in 1997 originally to represent the classical biomolecular structures in terms of this type of network diagrams. And this actually goes back to the original network models theory of Paul Fleury, talking about polymer physics, how it has been influenced our field. According to the polymer theory of polymer network, theory of elasticity of polymer networks, you represent the big structure, the macromolecule, in terms of nodes and springs. Nodes, in the case of proteins, were residues. Now we are talking about gene loci. And then springs refer to contacts, what is being measured by high-C experiments. Nodes undergo Gaussian fluctuations about their mean positions, and so do the internode distances. They also fluctuate Gaussianly. And you know, the position or the distance between two nodes obey this type of Gaussian distribution. Okay. That's the basic theory. For such a system, we don't have any specific interaction, energetics. We just take a rigorous account of the overall topology of the structure in terms of a network, elastic network, such that the free energy is purely entropic, and can be expressed as a harmonic potential, actually. So in the chemistry community, they like to call it, harmonic potentials. In the case of solid-state physics, we like to talk about forces. There was this special issue of Science in 2018 talking about how forces are behind form dictating the function, actually. So here I just put a parameter Aij. In the case of proteins, we have been using the same force constant for all connections. In the case of high C data applied to the genome, we are using the data that have been collected, which means the strengths of interaction association between those gene loci, or the probability of association. Rouse model, everybody knows, I guess. It's just a linear chain, which you can, the connectivity is represented by this tridiagonal matrix, and the total potential is written as a sum of harmonics. In the case of our approximation, instead of, the total potential, you can always write the total potential as a pre and post multiplication by the fluctuation vectors of the individual nodes. And this connectivity matrix just in this product gives yields the overall potential. In the case of generalization now to any network, you're going to have not only a linear chain, but off-diagonal terms also in the connectivity matrix. And the way you define those, there are, in the case of proteins, we analyzed back in 1997, we analyzed PDB structures and determined what would be the first coordination shell, etc. In the case of the high C data, we don't need to bother about that. We directly use the data that have been measured. Now, another just basic thing, if you are trying to calculate the covariance between the fluctuations of nodes I and J, you can write it as a Gaussian integral. The most important thing is that it is the covariance is just scales with the inverse of the connectivity matrix. It's a pseudo inverse, but physically what it is, this is what it is. And so we have this type of representation. In principle, the covariance is the reciprocal of the connectivity matrix, or vice versa. Okay. Now, in the application to the 3D genome, this is a recent review that will soon appear, I assume. It is, we take the contact maps, we represent them by this Gaussian network model, and then we calculate the square fluctuations of the gene loci. We compare with experiments. Anything that we do would be really, it's a very, very simple model, right? And it would be, to us, the most important thing is to see that first, it correlates with experiments. And secondly, we can make new predictions, generate new knowledge. So that's why we've been always comparing with experiments. Now we can, do this type of eigenvalue decomposition, obtain clusters of, or territories, if you like, of the chromatin. And we can compare them to TADs, the topologically associated domains or compartments, or, this hierarchical level of organization. We can calculate cross correlations and relate them to Chia-Pet experiments, for example. So these are all the things that we have been doing. Essentially, everything boils down to this very simple expression. The inverse of the connectivity matrix, but connectivity, it's not a binary. It is weighted by the strength of interactions from high C data. The inverse, ijth element, refers to the covariance of the nodes I and J, gene loci I and J. They tell us how much those two gene loci are correlated. And then the diagonal term will tell you about the fluctuation, size, okay? Motion, spatial displacement. Now we have not only a 3D structure, but we have also the equilibrium fluctuations, right? So this was the first paper actually we published in this area. And this is the result for one of the chromosomes where we have compared the mean square fluctuations predicted by the Gaussian network model with two different sets of experiments, attack-seq and DNA-seq. And I need an experimentalist to explain probably better why our predictions agree better with DNA-seq experiments. Always, apply to different cell types, all chromosomes, etc. But these are the two sets of experiments, after all. And you can see in these calculations, each node represents 5,000 base pairs, if you wish. And then the chromosome 17 is such that it is represented by, in this case, by 16,000 nodes. Nowadays, it's very easy. We do the calculation for the entire chromatin for 3 billion nucleotides represented in terms of those loci. So it is mathematically rigorous, exact, the solution. And then when we did this not only for chromosome 17, but for all other chromosomes, these are the correlations we get. As I mentioned, the correlation with attack-seq experiments also on accessibility of the gene loci is around 0.66. The one with DNA-seq is 8.81. And about this, you can see what happens with all chromosomes for that particular cell line. Interestingly, the two sets of experiments don't show such a high correlation as the correlation between theory and DNA-seq experiments. So yes, Gaussian approximation is an approximation, but it provides a good representation of the experimental data. More comparison with experiments. These are, now we are talking about compartments, much bigger entities. A and B, and A is divided into A1, A2, B is divided into B1, B2, B3, B4, for those of you who are familiar. And examining within those compartments, what are the square fluctuations of the nodes. Okay? So A1 fluctuates a lot, they are more exposed, etc. You can see the trend, the mean values and variances. These are experiments. This is theory, GNM. And again, among experiments, we agree much better with DNA-seq, not necessarily with attack-seq, and the two experiments themselves are not so compatible. So much about another comparison with experiments. So I'm just giving you some results to show that it is simple, but it works. Now, what is Chia-Pett experiments? What they do in those experiments, and they call them long-range correlations, actually they are not such long-range correlations because we are talking about things that are separated by 100 kilobase pair. So if you have a representation, each node representing 20 kilobase, now we're talking about 5 of them only, out of 1,600, as we saw in chromosome 17. So they are really very, very close to the diagonal, and it is very hard to observe. This is the typical high CMAP, and when you enlarge a region, they are observing things that are very close to our standard here, 100 kilobase pair. Then what we've done is, okay, we have those data for correlated pairs, and then others separated by the same distance, which are not detected to be correlated to those genes. Yeah. Oh, really? Are you sure? Are you sure? Because I started very late. Okay. I'm not even midway. So we have, if you compare with Chia-Pett experiments and then the background, you can see that what is being measured to be more correlated shows a higher covariance It's not perfect, but at least quantitatively we are at the right position. Now let's talk about the variability of the genome a little bit. Now we have the same DNA sequence in all cell types, but they differ from, they have different gene expression profiles. And for example, the TOTs are clustered in different ways. And I'm sorry, I have to move faster. And we have the same DNA sequence, but as we can see from the high C maps, there are structural heterogeneities. I'm not talking about the same type of cell structural heterogeneity, but heterogeneity across different types of cells. And we did the analysis for 16 different cell types. And we focus, in order to see better the differences, it is good to do an eigenvalue decomposition and focus on the dominant features. I'm going to skip this mathematical part to get to the interesting biological part. But the idea is normally, for 3 billion base pairs, you would have normally 60,000 nodes if you were using 5 kilobase pairs. Now we are doing the calculation for the entire chromatin, and we have seen that the top 500 are sufficient actually to represent the entire behavior. This is the high C map. This is the covariance map that we obtain, shown only for one of the chromosomes, just for simplicity. And the same thing, if we recalculate using a subset of modes, let's say 10 modes, we have this, we can discern now larger scale cooperativity. If we take 100 modes, we can observe smaller regions. And what we have shown in previous work is that approximately if you use 35 modes, you are exactly capturing the TAD size, topologically associated domains. If you take 5 to 20, this corresponds to compartments. So by looking at different windows, frequency windows, it is possible to see that it's not only TADs and compartments, but there's a whole hierarchy of organization in the genome. Different cell types, you can see they have different behavior. And then when we do the cross correlation between those mean square fluctuations, cell type, and then average over all columns, the average correlation is about 0.63. So that's how similar different cells are. And we wanted to see what's the origin. Is it because when we decompose, we have eigenvectors that give you the shape of fluctuation of the gene mobility profiles. And then you have eigenvalues, which is the prior probability or frequency. Okay. And then when we did that, we could see that there were swaps, actually, when we compare different types of cells to their modes. So if we swap the orders, if we get rid of frequency differences, we can see that we reproduce almost perfectly the same profile for all cell types. What does it tell us? It tells us that actually, if you don't have those differences in prior probabilities, you have practically the same behavior with a correlation of 0.85 across all cells. All cells possess the same repertoire of motions. Some of the motions are expressed, others are not. That's the whole point. And I will and based on that, you can construct for each cell type a differential mobility profile for the genes we call the MG. So from the average, from the individual profile as a function of gene loci, mean square fluctuation, you subtract the average. Now you have magnified differences. And those differences, now we're going to use them to screen, to compare with the DEGs. DEGs is differential expression of genes. We have this database that we can use for data for 125 different cell types. And then if we consider different 125 different cell types, and we use their known DEGs, expression profiles, and compare with the mobility profile of one of them. The question is, which particular cell's expression profile is most similar to the mobility profile of our queried case. And we can capture the same cell type, which means among 125 different gene expression profiles, we're using as filter our mobility profile for a particular type of cells, and we are observing that it is most similar to indeed that the expression profile for that particular site. So I'm finishing now. Differences in chromatin, they determine the cell differentiation we have done for many different cell types. The conclusion is that we consider these to be the first steps toward 4D genome. Good agreement with DNA, all those data. And as I mentioned before, some modes are silent. They are not being expressed. It is a little bit similar to what DNA does. You know, we say in different cells, in different tissues, some proteins are expressed, others are not. And this expression level is directly related to their mobility profile. And some gene loci moves or have the energy ability to undergo changes, others don't. And the dynamics is a determinant of cell-specific genome expression signature. That is from one of the models, 3D models, using elastic network models, just the softest modes to show how the entire chromatin moves. We have again, we're using population, high-C data, not single cells. But after all, the ingredient of the theory is the probability of contact. So we're happy with population average behavior. It is possible to carry out cell-specific mobility profile calculations using data. And then this database has been put together very recently by Eres, Lederman, Aiden, and others, where they have now compiled the models for the chromatin. So we can serially apply the anisotropic network model to generate this type of movies and see what is the most robust. Last word about the GNM. It is approximate. It lacks energy. It lacks structural complexity. It is purely based on contact topology. It gives information on entropically driven correlations. I think the physical approximation is being somehow compensated by its mathematical rigor, I would say. We have a unique solution that takes account of the entire topology, and especially when we look at the global features, they are very robust and give us information on hierarchy, cross-correlation, and specific mobility profiles. And with that, I thank you for your attention. It's not a good thing to be the last speaker, especially when during the day we have been lagging behind schedule. And I hope it wasn't so bad as the last talk.